Okay, hello. This is the second lecture, Zoom lecture for you guys. This is a combination of lectures 11 and 12, aerobic gram-negative rods 1 and 2. And the information on these lectures or this big lecture will not be tested until the week after you return from vacation. So if you want to do your homework, don't do it after your vacation. You can submit it during the weekend or definitely the Monday after you return. All right, let's get started. 150 or so slides. Okay, so I'm going to be talking about the non-fermentative aerobic gram-negative rods, and it includes Pseudomonas aeruginosa. That's one of the biggest organisms in this group. Pseudomonas fluorescence and Pseudomonas Maltophilia. That organism has an identity problem. It used to be Pseudomonas Maltophilia, then it went to Xanthomonas Maltophilia, and then now it's Pseudomonas Maltophilia. I'm going to talk about Moraxella species, Moraxella lacunata. Another main Moraxella species is Moraxella branamella cataralis. Or Moraxella species is generally a pathogen that infects the upper respiratory areas. Acinetobacter species is another non-fermentative gram-negative rod. It utilizes carbohydrates differently. It definitely does not ferment. Alkaligenes faecalis is another non-fermentative gram-negative rod. I'm going to talk about Chrysotobacter meningiosepticum, Burkholderia cepacia, Bartonella species. There's several Bartonella species that causes trench fever, etc. And another type of cat scratch fever. All right, family Legionella, okay, Legionella pneumophila, okay. 85% of documented legionellosis of Legionella causes legionellosis. There's several serotypes, 14 serotypes, 16 species, and 21 serotypes. So there's a lot of organisms that are in this group. There are different serotypes and different species. I'm also going to be talking about Nyseria species. Nyseria gonorrhea, meningitis, Lactomica, Zika, and Branhamella cataralis. Branhamella also has an identity problem. Excuse me, that used to be called Branhamella cataralis used to be called Nyseria cataralis, or even Moraxella cataralis. It used to be, it's now Branhamella, used to be Moraxella, and originally used to be Nyseria cataralis. These organisms are gram-negative rods or coccobacilli. They're strictly aerobic and they're modal. If they're modal, it has polar flagella as opposed to Enterobacteriaceae when the flagella are peritrichous. These organisms are incapable of utilizing carbohydrates. If they do, they're oxidizers. They're not fermenters. Remember the Pseudomonas aeruginosa on the TSI is K over K. Well, these organisms are K over K on the TSI. They're oxidative rather than fermentative. They're non-glucose fermenters, like I said, K over K. Pseudomonas species either grow at 25 degrees or they grow at 42. 37 is not optimal. Okay. Pseudomonas aeruginosa is the most frequently isolated pseudomonad. Okay, it's the most frequently isolated. Pseudomonas aeruginosa is common in burn victims and also to cystic fibrosis. Pseudomonas, if it's in cystic fibrosis, it can be very, very problematic. One reason, because if you work in a children's hospital and you have cystic fibrosis, one of the common culture characteristics of Pseudomonas organism from a cystic fibrosis patient is that it is super mucoidy. It's really, really goopy, okay? And so in order to work it up, it's Pseudomonas, it's oxidase positive. That's basically the only thing you can do. It's an oxidase test, but you need to isolate colonies if you're going to be doing sensitivities. So you have to subculture it and subculture it and subculture it daily to get rid of the capsule. It's a highly capsulated organism. It's very, very mucoidy. And once you get rid of the capsule, then you can further do workups and do your sensitivities. But that's one of the characteristics of Pseudomonas aeruginosa from cystic fibrosis superinfection. Ear infection. Pseudomonas aeruginosa is commonly found in swimmer's ear. Make sure you know this. Ear infection, swimmer's ear. Okay, this is one of the diseases about Pseudomonas aeruginosa I'll be asking you about. Also, two burn infections and cystic fibrosis. Those are the three main ones for Pseudomonas aeruginosa: burn infection, cystic fibrosis, and swimmer's ear. Necrotizing keratitis. A lot of things cause necrotizing keratitis, but Pseudomonas aeruginosa can be implicated, causing necrotizing keratitis. IV drug users. One of the nosocomial infections. One of the nosocomial causes of nosocomial infections, fatal lower respiratory tract infection. So lower respiratory tract infection is below the bronchus. It's the lower parts of the lung. Okay, and when it gets that, when the infection gets to be that bad, that kind of infection is seen in immunocompromised patients. Pathogenic factors include exotoxin A, alginate, proteolytic enzymes, pili, and endotoxin. I'm not going to be talking or testing you on the pathogenic factors of Pseudomonas aeruginosa. Laboratory characteristics, you know it's a gram-negative rod. You know it's aerobic, okay? It's oxidase positive. It oxidizes the glucose. It's modal. It has polar flagella, and it does reduce nitrate to nitrite, but it doesn't belong in Enterobacteriaceae because it's oxidase positive, and it has polar flagella. Polymyxin susceptible. Pseudomonas aeruginosa is polymyxin susceptible, and it likes moist areas. Okay, wherever moisture accumulates, Pseudomonas aeruginosa will be isolated there. Like, for example, the swimmer's ear, the tracheotomies, indwelling catheters, weeping cutaneous wounds. So those are sites of patients who are like in long-term facilities that those are the moist areas: the ear, the trachea, indwelling catheters, and weeping cutaneous wounds. Pseudomonas aeruginosa likes to grow at 42 degrees. Okay, so you need to distinguish that between Campylobacter, because Campylobacter also grows at 42 degrees. And the way you can distinguish between Pseudomonas aeruginosa and Campylobacter is the gram stain. Campylobacter are thin gram-negative rods. Remember the gram-negative seagulls? Pseudomonas aeruginosa are fatter gram-negative rods. And also, too, the smell. Pseudomonas aeruginosa has that fruity odor smell, and Campylobacter doesn't. And what else? All right, so growth at 42 degrees. This, you see a plate like this, you see that metallic sheen, and it's beta hemolytic on blood. That's classic Pseudomonas aeruginosa on blood. Nice shiny colonies. The appearance, make sure you know the appearance, okay? I hope you're collecting different things like diseases and appearance and colony characteristics. Okay, so I gave you that large comprehensive, matching comprehensive. Go through that and look at Pseudomonas aeruginosa, and there's colony characteristics. Pseudomonas aeruginosa has resembles fish scales, ground glass appearance, and it resembles fish scales. Make sure you know that descriptive term. So if you see ground glass appearance resembling fish scales, that's Pseudomonas aeruginosa, and that's what it looks like on blood. And it is beta hemolytic. It has that grape-like or fruity odor. On McConkey, it's colorless and it's flat and it's regular. So it's a non-lactose fermenter because it's colorless. HE, it's inhibited, won't grow, pigment producer. Okay, know the pigments of Pseudomonas aeruginosa. It has pyocyanin, which is the blue. And then it also has pyoverdin or fluorescein, yellow. Okay, pyoverdin or fluorescein, it's yellow. So Pseudomonas has the blue pigment and the yellow pigment, pyoverdin and fluorescein. The next Pseudomonas is Pseudomonas fluorescence. It's an uncommon, it's less common than aeruginosa, found in urinary tract infections and the respiratory tract and wound infections and bacteremia. Now, the pigment for fluorescence is the yellow pigment, fluorescein or pyoverdin, so not pyocyanin. So aeruginosa is pyocyanin and the fluorescein or pyoverdin, so it can be yellow or green. Fluorescence is yellow only, the fluorescein and pyoverdin. Fluorescence does not produce pyocyanin, like I just said, and it doesn't grow at 42 degrees. Okay, this Stenotrophomonas Maltophilia, also known as a long time ago Pseudomonas Maltophilia. This organism can be problematic because it's an MDRO. MDRO, it stands for multiple drug resistant organism. It's one of those organisms that's difficult to grow and not difficult to grow, but difficult to treat because it's hard to find antibiotics to treat it. It's kind of like MRSA, all right? But if you do, it can be a contaminant. It's found in debilitated patients, wound infections, bacteremia, pneumonia, and urinary tract infections. Formerly Pseudomonas, formerly Xanthomonas. In contrast to Pseudomonas aeruginosa, like I said, this used to be a Pseudomonas, it's oxidase negative. Classic test for Pseudomonas aeruginosa is the oxidase test. It has that fruity odor, oxidase positive, okay? And the Maltophilia is resistant to most anti-pseudomonal drugs. Moraxella lacuminata is normal flora of the mucous membranes. However, if it can be opportunistic, I knew somebody who had a Moraxella infection. It just colonized the nasal tract, and she just constantly had infections. So it's part of the normal flora of the mucous membranes, and it can also cause conjunctivitis. Moraxella lacuminata is a gram-negative coccobacillus, coccobacilli. This one is oxidase positive, and this organism does not grow well on McConkey. So other than really the Pseudomonas aeruginosa, it will grow on McConkey as a non-lactose fermenter. Generally speaking, a lot of the non-fermentative gram-negative rods do not like to grow on McConkey. Acinetobacter species. This is an organism that's difficult to treat. You can have saccharolytic, which means it lyses sucrose, asaccharolytic, non-hemolytic. The different combinations, saccharolytic, non-hemolytic, saccharolytic, hemolytic, asaccharolytic, non-hemolytic, and asaccharolytic, hemolytic. You're not going to need to know those. It's just that there's different categories of Acinetobacter species. The ACB complex stands for Acinetobacter calcoaceticus, and Acinetobacter genomic species. So I'm not going to test you on these complicated subspecies and the ACB complexes. It's just that the different characteristics of this type of organism. However, Acinetobacter is important because it may colonize moist skin. So remember, Pseudomonas like moist areas. Acinetobacter likes moist areas too, like the skin, the genitourinary tract, and respiratory tract. Again, it's also common in nosocomial infections, compromised patients who are on the respirator. And just like the Stenotrophomonas Maltophilia, Acinetobacter is also considered an MDRO. It's resistant to most antibiotics. So if you have Acinetobacter species, it can be a nightmare in trying to treat the infection. Laboratory characteristics is the gram-negative coccobacillus. Sometimes it may appear diplococcoid. Don't focus on that. The only diplococcoid you need to know are like pneumo. And upcoming, there's a gram-negative diplococcoid coccus that's really important to know. Pneumo, as you recall, is gram-positive diplococci. Acinetobacter, like Stenotrophomonas Maltophilia, is oxidase negative. Okay, and it's non-modal, it's nitrate negative. This one will grow on McConkey, and it will oxidize glucose and lactose. There's that saccharolytic and asaccharolytic again, and you distinguish that by glucose oxidation. When we do the review, I know there's a lot of information I'm throwing at you on Acinetobacter and in all of these organisms, the Pseudomonas, but I'm going to pick it even further and direct you into what information is important and not important. This is another one, Chrysiobacter meningiosepticum. Natural habitats found in soil, plant, water, but not humans. It's important because it causes nosocomial infections. And in the hospital, it can be isolated from nebulizers, water baths, and saline. So a lot of these non-fermentative gram-negative rods are problematic organisms that cause nosocomial infection. Chrysiobacter meningiosepticum can survive in tap water, chlorinated tap water. Okay. Pathogenicity, neonatal meningitis, and bacteremia. And several epidemics with high mortality rates in hospital nurseries have been caused by Chrysiobacter meningiosepticum. So it's not an organism that we can overlook, but I want you to at least know it does exist, and it can be found in premature babies. The way this organism looks, it's long, thin, with bulbous ends. It almost looks like a peanut shape. It has bulbous ends, okay? If you look at it, if you have an imagination, it kind of looks like peanuts. This one is non-modal, and it's oxidase positive. So keep in mind that when I do these exams, that I use pictures, and the pictures will be used. I've decided to use them for your lab grade, okay? So if you see these bulbous ends, these rods with bulbous ends, that's Chrysiobacter meningiosepticum. So start making a picture of a collection of the different pictures that are in these lectures. Another unique characteristic about meningiosepticum, Chrysiobacter meningiosepticum, is that it produces pigment. Now, this actually kind of looks like micrococcus. Remember, micrococcus is bright lemon yellow. Well, this is not bright lemon yellow. It's light yellow to orange, okay? So meningiosepticum is light yellow to orange. That's the color. If you see light yellow to orange versus bright lemon yellow, light yellow to orange is meningiosepticum. Okay, variable growth on McConkey. Again, these organisms have trouble growing on McConkey. Chrysiobacter meningiosepticum is indole positive, and the pigmentation is unique that it can be used as reference. Alkaligenes faecalis, it's most often a contaminant as well, typically found in water and soil, and can cause opportunistic infections. Respiratory and urinary tract and blood. Nosocomial infections via respirators, hemodialysis instruments, and IV systems. It's a non-lactose fermenter, aerobic and oxidase positive. Alkaligenes faecalis does not oxidase glucose. It has positive, it's modal. This one, this non-fermentative gram-negative rod has peritrichous flagella, and it does grow well on McConkey, but it's not an Enterobacteriaceae because it doesn't oxidize glucose. It's oxidase positive. I'm sorry, it's oxidase positive. Okay, Burkholderia cepacia. Burkholderia cepacia is another interesting organism. This organism, like Pseudomonas aeruginosa, is also can be found in patients who have cystic fibrosis and chronic granulomatous disease patients, urinary tract infection and respiratory tract infection. So right off the bat, it's similar to Pseudomonas aeruginosa. It has green coloration on chocolate. This is Burkholderia cepacia, oxidase positive, motility positive. So it is kind of similar to Pseudomonas. Okay, grows in McConkey, aerobic. PC is plate count media, OSBL. BCSA is Burkholderia cepacia selective agar. So there's different types of media that you can use other than the conventional blood chocolate and McConkey agar that you can use to isolate Burkholderia cepacia. Pathogenicity. The next organism is Burkholderia pseudomalii. Okay, first one is cepacia. Next one is Pseudomalii. Pseudomalii is a possible bioterrorist weapon. The bacteria. It's oxidase positive, motility positive, and this organism. So I'm going to talk about Pseudomalii more later on, and then Burkholderia malii. Okay, there's Pseudomalii and malii. Malii is found mostly in horses, and it causes a disease called glanders in horses. So make sure you know the disease glanders. That's going to be on a test in horses, okay? If you see horses, Burkholderia malii. If you see glanders, a disease in horses, that's going to be on the test. I'll go over that when I do the review. I'll go over this again during the review. Malii, Burkholderia malii is oxidase positive. It's non-motile. This one likes to grow on McConkey, and it's aerobic. Okay, biochemical identification. The Pseudomonas, usually definitive ID of Pseudomonas, used mostly, I didn't forget, used mostly for a definitive ID of Pseudomonas aeruginosa, motility, and the ability to produce cytochrome oxidase. Pseudomonas is really easy to identify. You saw what it looked like on blood. You can smell it, the fruity odor, and it's oxidase positive. If you see it, and it grows at 42, if you see those characteristics, it's pretty sure you can be pretty confident that you have Pseudomonas aeruginosa. Identification of non-fermenters. The identification is initial assessment includes the gram stain oxidase motility testing. To do the motility testing, you're going to probably stab an OIM tube. Hopefully, we will get those in the laboratory so you can see how an OIM tube works. And then there's various identification kits and systems that you can use. Pseudomonas, look for growth at 42 degrees. Aeruginosa will grow at 42 degrees. And the pyocyanin production, that's the green, okay? Pyocyanin production versus the fluorescein and the pyoverdin, which is yellow. Denitrification. More on the ID systems, the cetramide agar, which we won't be using. Pickett's fluorescence, lactose and nitrate medium. And that one produces fluorescence, and you look at it under black light so you can see the pigment. Another one is Seller's medium, Pseudomonas aeruginosa, Acinetobacter, and alkaligenase. I'm not going to get into these various media and identification systems because, even since I got off the bench, I'm sure more identification systems have come out on these non-fermenters. More identification procedures like the API, Enterobacteriaceae uses the 20E. For the non-fermenters, it's API NFT, non-fermentative test. There's the oxiferum tube, the non-fermentative system by Remmel, and then there's a crystal enteric non-fermenter system. Okay, it looks like a mess, but this is a flow chart for the gram-negative facultative gram-negative rods. I'm not going to test you on this. Basically, I'm giving you the information now. I'm going to probably reorganize this, but I'm not going to test it on this flow chart. Okay, Legionella causes Legionnaire's disease, pneumonia with a high fatality rate of 10 to 20%. Epidemic and nosocomial infections. So Legionnaire's disease came about with when there's an American Legion convention, and this convention was like on a cruise liner, and all of a sudden, you know, a whole bunch of members of the American Legion got sick, and they came down with pneumonia, and they couldn't find out for the longest time what was causing their pneumonia until they finally isolated it, and it came from the air conditioning system. Another reason why it was difficult to identify Legionella was because this organism is difficult to stain. Okay, so Legionella causes Pontiac fever. So make sure you put that disease in your collection of diseases. And it's caused by Legionella. The organism is called Legionella pneumophila, okay? Acute non-pneumonic, febrile illness, self-limited, and it's non-fatal. So all these people from the American Legion, they got pneumonia, but they didn't die. They just got really, really sick. Okay, characteristics, it's ubiquitous, meaning that it's all over, it can be found all over us, all around us. Primary aquatic saprophytes, air conditioning units. So the major reservoirs for Legionella could be warm freshwater sites or air conditioning units. Stored potable water also can be found in lakes, rivers, and marine waters. Legionella is aerobic, and on the gram stain, it's a thin gram-negative rod. So that's why it was difficult to see, because it's a thin gram-negative rod. And like I mentioned in the last lecture, one of the techniques that you can use is when you do the gram stain, just stop after the crystal violet and iodine, and then don't decolorize it. Just leave it as is, rinse it with water, and look at it under the microscope, and then you'll see these gram-positive. Well, I shouldn't say gram-positive, but these dark staining rods that are actually there, which if you take the stain all the way through, but decolorizing and backstaining with the safranin, counterstaining with the safranin, then you'll get the gram-negative rods, but they are difficult to see. Legionella can also be small bacilli, coccobacilli. Very, very difficult to see. These are pleomorphic filamentous rods, 100 microliters in length, and it's fastidious, meaning that it has special nutritional requirements. It requires L-cysteine and iron for nutrition. L-cysteine and iron is a nutritional requirement. It's usually motile. The cell was distinctly unique branch chain of cellular fatty acids. Respiratory, Legionella, from the infection with the American Legion individuals that came down with pneumonia. Respiratory tract secretion is a specimen of choice. Fluids, especially sputum, is a specimen of choice. If you suspect Legionella, you transport it to the lab as soon as possible. If you can't culture it, then you can hold it up to 48 hours at refrigerated temperatures. If longer, then you freeze it at not 20 degrees, but it's minus 20 degrees centigrade. I'll change that. Because it's difficult to see, you can use direct fluorescent antibody, DFA, or special stains like Giemsa or the silver stain. Okay, the media of choice. I know I mentioned nutritional requirements, but for Legionella, okay, it's buffered charcoal yeast extract. Legionella is BCYE. BCYE is for Legionella, buffered charcoal yeast extract. Make sure you know that, and that's a testable item. Legionella can also be found in cystic fibrosis, and if it is in cystic fibrosis, again, like I said, the specimens are usually very mucoidy, and you can add acid for better recovery. If you have tissue, then you may consider homogenizing the tissue before you plate it. For culture, it's more than 24 or 72 hours. You want to keep it for 2 weeks. 35 degrees incubation, CO2, and with 80 to 90% humidity. Okay, so remember, Legionella is buffered charcoal yeast extract. The colonies will not appear until 3 to 4 days or 5 days. Okay, the colonies will appear. And colony morphology also is characteristic for Legionella. And make sure you know this, it's cut glass morphology with internal granular speckling. So cut glass morphology. Okay, remember Pseudomonas Aeruginosa was fish scales. So this one is cut glass morphology. Start collecting as you go through these notes and listen to the lectures. Colony morphology, you're going to be, it's going to be on the matching, where I'm going to give you various morphologies and you're going to tell me what organism that morphology belongs to, like fish scales, or cut glass, or railroad tracks, or mercury droplets, which I haven't talked about yet. Okay, so start collecting those. Legionella identification to species level is not important. All the doctor needs to know is Legionella, and then he'll know how to treat it. Sensitivity testing, you don't have to do sensitivity testing. Like I said, the doctors already know how to treat Legionella. Okay, that's it for Legionella. Nyseria, okay, pretty important group of organisms. These are gram-negative cocci that occur as diplococci. So these are gram-negative diplococci. Okay, we just had gram-positive diplococci. That was your pneumo. But if you see gram-negative diplococci, now you need to start thinking about Nyseria. Nyseria is important because that's the one that causes gonorrhea. The way it's shaped is that it's kidney bean shaped, and these gram-negative cocci kind of are paired. And if you recall, the pneumococci are lancet shaped, so the points come together like this, points. But for Nyseria, they come together like this, okay, like two kidney beans that are stuck together just like that, and that's their configuration. They resemble kidney bean shape. The flat side is attached to each other just like this. Okay, if you see that gram-negative diplococci, that's Nyseria. You do an oxidase test, it's positive. So then you got something serious you're going to be working up. Fastidious, this requires blood as a source of iron. It also needs to be incubated in CO2. This organism, like I said earlier, is oxidase positive. So gram-negative diplococci, oxidase positive, likes to grow on chocolate. The media of choice for Nyseria is modified Thayer Martin. I'll mention that later. Okay, it's the etiologic agent for GC, or the clap. Okay, anatomic sites to identify possible Neisseria gonorrhea infection are the urethra, anal, conjunctiva, vagina, oral, joint, endocervix, and prostate. Joint is in there because Neisseria gonorrhea has a tendency to colonize in the joints. So if you can have an athlete may not have a venereal type infection, but Neisseria gonorrhea can have a tendency to colonize the joint area. So synovial fluids, if you're culturing a synovial fluid, the doctor's trying to see if there's a Neisseria colonization in the joint. Conjunctiva, just like I mentioned, can affect the eyes. Purulent arthritis, again, back to the joints and the synovial fluid. Neisseria gonorrhea has a tendency to colonize the joints. So purulent meaning a lot of white blood cells, that's causing the individual to have arthritis. All right, for the Neisseria species, make sure you know the carbohydrate patterns. So for Neisseria gonorrhea, that's the first one I'm going to talk about. It goes glucose, maltose, lactose, and sucrose. So for Neisseria gonorrhea, it's glucose positive. Okay, so one way to remember that is G goes with G. Neisseria gonorrhea is glucose positive. Neisseria gonorrhea will not grow on blood, like I said. It loves chocolate, specifically modified Thayer-Martin, MTM agar. Okay, so those are down below are the different types of Nyseria gonorrhea agar that you can use. Modified Thayer-Martin, Martin-Lewis agar, Gembec is a special identification system, and New York City agar. So these media are selective for Nyseria gonorrhea. And this is what it looks like on modified Thayer-Martin. If you look at it closely and you look at and compare to the other species of Nyseria like Meningitis, like Lactomica, and Zika, look for similarities or differences. Media for Nyseria gonorrhea, like I said, modified Thayer-Martin. It consists of colistin to inhibit gram-negatives, nystatin to inhibit yeast, and vancomycin to inhibit gram-positive organisms. Trimethoprim sulfa, trimethoprim, which is a sulfa antibiotic, will inhibit swarming proteus. Martin-Lewis media has antifungal, so that yeast will not grow on that media. Gembec contains sodium bicarbonate pill, and that creates a CO2 condition. And NYC agar, Mycoplasma ureaplasma will also grow, but it's still selective for Nyseria gonorrhea. This is the Gembec system. So that media there is the modified Thayer-Martin. And if you look to see where that white pill is, so it has a little compartment there where you drop a white pill, you add water to it, cover it with the lid, and that pill creates a CO2 atmosphere. And that's how it's transported in that Ziploc bag. Neisseria meningitidis, the next one, fulminating meningitis. It's really bad. But it may be, meningitidis may be part of the normal flora in your nasopharynx. And that's called non-pathogenic Neisseria. So if you do a sputum culture, you're going to see that on blood and chocolate, you're going to see a whole bunch of different colonies. And if you swipe and you take a random swipe of all the colonies, like in a primary sweep, and you do an oxidase test, you're going to see oxidase positive. And then you're going to say, oh my God, that's non-pathogenic Neisseria. Does that mean, am I going to come down with meningitis? No, it's not. It's part of your normal flora. So that's non-pathogenic Neisseria. And characteristics, colony characteristics is very similar to Neisseria gonorrhea. The gram stain is identical to Neisseria gonorrhea. Okay, and this is what Neisseria meningitidis looks like on Thayer-Martin. Okay, see if you can try to point out any kind of differences as compared to Neisseria gonorrhea. So carbohydrates, remember, glucose goes with Neisseria gonorrhea, goes with G. So if you go in order, glucose, maltose, lactose, and sucrose, Neisseria meningitidis is positive for both glucose and maltose. So meningitidis is positive for glucose and maltose. M goes with M, glucose only for Neisseria gonorrhea, and lactose and sucrose are negative. Okay, so you kind of see a pattern there. Neisseria lactomica is an organism that's normal flora found in the oral pharynx and the nasopharynx. Usually is avirulent, meaning that it's not aggressive. Rare cause of bacteremia, endocarditis, and meningitis. So it's a Neisseria species, but it's not really a significant pathogen. It's very similar, colony morphology is similar to Neisseria gonorrhea, and it will grow on modified Thayer-Martin chocolate, and this one will grow on blood. Remember, Neisseria gonorrhea does not like blood. And there is Neisseria lactomica. You can point out any colony characteristics that are different than Neisseria meningitidis and gonorrhea. Let me know, because there are no differences. All right, carbohydrates. Okay, glucose is for gonorrhea, maltose is for meningitidis, lactose positive for lactomica. Okay, right? Lactose positive for lactomica. So there's a pattern there. Okay, finally, Nyseria Zika, found in the oral pharynx, usually avirulent, and this one's found in immunodeficient patients. found in abscess, bacteremia, and endocarditis. Colony is similar to Nyseria gonorrhea. This one likes chocolate and blood, okay? And there is colonies, so you can take these colonies or these isolates on modified Thayer Martin, and they all four of them look pretty identical. Okay, so Nyseria Zika is the weirdo. So Nyseria Zika is positive for glucose, that's fine. It's positive for maltose, and that's fine. And it's positive for sucrose, that's fine. But this time the lactose is negative. You would think that with Nyseria Zika and the four carbohydrates, that it would be all positive for all four. But for Nyseria Zika, the lactose is negative. Okay, so gonorrhea is glucose only. Meningitis is glucose and maltose. Lactomica is glucose, maltose, and lactose positive, and Zika is glucose, maltose, and sucrose positive, but the lactose is negative for Zika. All right, just remember, Nyseria Zika, lactose is negative. All right, Moraxella catarrhalis used to be called Branhamella catarrhalis, and it used to be called Nyseria catarrhalis. So this is normal flora, again, of the upper respiratory tract and the female genital tract. Causes ear infection, respiratory, like pneumonia, sinusitis, can be serious bacteremia, and meningitis. Moraxella catarrhalis. It is a significant organism. Gram-negative diplococci. Nyseria is gram-negative diplococci. Okay. Colony morphology is similar to Nyseria gonorrhea. Variable growth on Thayer Martin. Good growth on blood. And as you can see, looks like gonorrhea, looks like meningitis, looks like lactomica, looks like Zika. Okay, this is Moraxella catarrhalis. Carbohydrates. Moraxella catarrhalis is negative for all the four carbohydrates. Negative for glucose, negative for maltose, negative for lactose, and negative for sucrose. Positive nitrate, DNA is positive, and its beta-lactamase is positive. So you don't want to treat it with a cephalosporin. Okay. Moraxella is catalase positive. Oxidase positive. Differentiation scheme to identify Nyseria gonorrhea. You need to use enriched selective media. So usually it's the modified Thayer Martin is what they'll use. Martin Lewis, New York City agar, and Gembeck. Gembeck usually is used for transport. Thayer Martin and Gembeck contains Thayer Martin. In the microbiology laboratory, you should have Thayer Martin plates. So you take colonies growing from the Gembeck and you subculture it onto Thayer Martin plates. Okay. Martin Lewis and NYC agar are not typically used, but those are the two main ones. Modified Thayer Martin in the laboratory and Gembeck used as a transport. Remember, Gembeck is the one with the CO2 pill. To identify Nyseria gonorrhea, remember, Nyseria gonorrhea does not like blood, good growth on chocolate. And those are the four carbohydrate tubes. They don't use that anymore. If it's positive, it would be yellow. If it's negative, it would be red. So I really miss that four tube screen. I mean, you inoculate the four tubes, and then at the end of your shift, you look at the colors and you know what you have. So it's glucose, maltose, lactose, and sucrose in that order. So it's rarely used anymore, unfortunately. I like that system. And as you can see, all the colonies look the same. Okay. All the Nyseria and the Branhamella catarrhalis, or the Moraxella catarrhalis, they all look the same. Carbohydrate utilization, that's a quad form, uses the four carbohydrates. This is another system that's used. Again, if the glucose is positive, that means it's gonorrhea. If the glucose and maltose is positive, then it's meningitis. Glucose, maltose, and lactose is positive. You know the pattern, okay? So as you can see, C is control, G is glucose, M is maltose, L is lactose, and S is sucrose. So as you can see, for example, in lactomica, you see yellow under the G, M, and the L. Under gonorrhea, you see yellow under the G. In meningitis, you see yellow for the G and the M. Okay, so look at these closely. C stands for control, but you have your 4 carbohydrates there. Something like this, I can give you on a lab exam, okay, on a lab quiz. So make sure you know, make sure you're familiar with this, and you can see the positive reactions for the 4 carbohydrates, okay? Okay, so specimen processing directly inoculate chocolate agar in the modified Thayer Martin and transport to the laboratory, preferably in the GEMBEC where you have CO2 with the culture. Carbohydrate utilization, once you're in the laboratory, you're going to incubate in a CO2 incubator. And recommended use, the swab. Use a calcium alginate wool tip swab for collection. Calcium alginate wool tip swab for collection. And look at the colony morphology. Easy. I mean, you have those 5 organisms there, and they all look the same on the selected media, the modified Thayer Martin. It likes increased CO2. That's very important. And you see small, moist to mucoid, grayish white colonies. And there's a hyperlink there. You might want to open it up for further identification. So when you have gonorrhea, you need to, your laboratory, your microbiology laboratory should have a protocol on reporting a positive Nyseria gonorrhea, such as, and this is an example. So what you report out to the doctor after you call it, you put in the report, many PMNs and many gram-negative diplococci occurring both intra and extracellularly, morphologically resembling Nyseria gonorrhea. Okay, so morphologically resembling Nyseria gonorrhea. So it's kind of non-committal, but what you're doing is you're directing, you're giving the idea that all indications point to Nyseria gonorrhea. But for legal reasons, I guess it's better to be safe, morphologically resembling Nyseria gonorrhea. Only symptomatic male with urethritis. Nyseria gonorrhea is females are usually asymptomatic. Okay, the next organism is Bordetella. Bordetella is the organism that causes whooping cough. Whooping cough. So make sure you know that disease, Bordetella pertussis. The next one is Bordetella parapertussis, which causes a pertussis-like symptoms, and it's less severe than the whooping cough that is produced by Bordetella pertussis. And then the third species of Bordetella is Bordetella Bronchoceptica, found in respiratory infections in various animals, dogs, cats, and rabbits. Upper respiratory infection in immunocompromised patients, and urinary tract infection, meningitis, bacteremia, endocarditis. So this one is less significant. The most important one is Bordetella pertussis, followed by parapertussis, and then the Bronchoseptica. That one is, I mean, for animals, I'm sure it's important for animals, but not for humans, okay? Bordetella gram-negative coccobacillus occurs singly or in pairs, and it's also pale staining. It's difficult to see. Bordetella is obligate aerobe. For pertussis, it's among the most fastidious bacteria known. It requires nicotinamide, cysteine, organic nitrogen, and amino acids. So it has a lot of nutritional requirements. I'm not going to ask you what the nutritional requirements are, but the media of choice is called, and it's easy to remember because it's called Bordetella Bronchoceptica. B-O-R-D-E-T. It's part of the name, Bordetella Bronchoceptica. That's how it's pronounced. Or B.G. agar. Bordetella or B.G. agar. Another media of choice can be the Reagan-Low, but the Bordetella is easier to remember. So Reagan-Low or Bordetella. Modified Jones-Kendrick is another, can be another media of choice. Bordetella, no growth on infusion agar or blood agar. Okay, if you have a patient or you have a child, usually these are pediatric patients that have the whooping cough. The the doctor will say, rule out Bordetella. So right away, you're going to pull out your Bordetella agar. It's a black plate, and you just go ahead and plate your specimen onto the BG agar. So specimens for Bordetella, 2 nasopharyngeal calcium alginate swabs. Plastic will kill this organism, so make sure you know that plastic swabs or any kind of plastic will kill Bordetella. Okay, it's red there for a reason. When direct plating is possible, use Reagan and low media, semi-solid transport media. So Reagan low is used for transport, but in the laboratory, once it's in the laboratory, plate it onto a BG agar, the Bordetella agar. Also, too, you can use direct fluorescence antibody to see the organism from clinical samples. You incubate it at 37 degrees for 12 days. So Bordetella is a 12-day incubation, but you need to examine it daily. Again, no growth on blood, on primary isolation. Okay, so Bordetella, colony morphologies. Okay, so you've had different colony morphologies already, like fish scales, railroad tracks, okay, etc. This one, Bordetella pertussis, has mercury droplets, pearl-like luster resembling mercury droplets, and this is on the BG agar, okay, not blood because it doesn't grow on blood. This is on the BG agar, and you might want to look at page 485 for more explanation on it. But Bordetella pertussis has the mercury droplets. So put that description in your collection of colony morphologies, because you're going to have a section on matching on colony morphologies. And I'll mention that during the review when you come back. Okay, so colony morphology, whitish gray as it grows older. The colonies turn whitish gray as it grows older. And you want to examine the plate with a dissecting microscope to look at the mercury droplets. PCR testing for better specificity for Bordetella pertussis. A lot of technology in microbiology is turning towards PCR testing because it's more specific. Bordetella pertussis will not grow on McConkey. Okay, it's oxidase positive, so it won't grow on blood, it won't grow on McConkey. That's why if you know you have whooping cough, you need to have the Bordetella, the BG agar on hand. So it's oxidase positive, motility negative. Bordetella pertussis grows a little faster, visible growth in 1 to 2 days. This one will grow on McConkey, and it's oxidase negative. Okay, again, you're going to be putting this on BG agar, just as if you're going to rule out pertussis, Bordetella pertussis. Okay, visible growth sooner, 1 to 1.5 days. Good growth on McConkey, and it's oxidase positive, motility positive, and urease positive. This is Bronchoseptica now, not Parapertussis. So the third species of Bordetella is Bronchoseptica. This is the one, remember the animals? Okay, this one grows faster, 1 to 1.5 days. Likes McConkey, oxidase, motility, and urease positive. Okay, that's it for Bordetella. 3 significant organisms, actually 1 main significant one, the pertussis, then the parapertussis, and then the Bronchoseptica for animals. Don't forget that. Okay, this now is Brucella. 4 species of Brucella. Brucella melitensis, Brucella abortus, Brucella suis, and Brucella canis. Okay. Etiologic agent of brucellosis. And this is an obligate intracellular parasite which can cause epizootic abortions in various animals. Okay, various animals, usually the farm animals like cattle, pig, sheep, dogs, and horses. These are, Brucella are the farm animals, okay? Cattle, pigs, sheep, dogs, and horses. Brucella is the organism that caused the milk industry to pasteurize its dairy products, okay, because of the Brucella infections. And extensive compulsory vaccination program for susceptible animals, not humans, but vaccination for the livestock, the cattle, pigs, sheep, dogs, and horses. Okay, those are the animals that are being vaccinated. So pasteurization of the dairy products was brought about because of Brucella outbreaks. Clinical significance, it occurs in patients with occupational or vocational exposure to animals or laboratory cultures, like individuals who work in butcher shops. Okay, and so you can probably pick up a Brucella infection if you work in a butcher shop or a slaughterhouse. The disease is transmitted to man by direct contact with infected animals through ingestion of contaminated dairy products. Another disease that you need to collect is Undulating Fever or Maltophilia. That's caused by Brucella. Causes arthritis, spondylitis, endocarditis, lymphadenopathy, and splenomegaly. So that's undulant or Maltophilia. Brucella is a gram-negative coccobacillus, difficult to see, and it's pale staining. It's non-modal, it's an obligate aerob, and it likes CO2, and it's a slow grower. Usually, typically for Brucella, you want to keep the culture for 30 days. Okay, so Brucella, laboratory safety, it's a class 3 pathogen, so it can be used for a bioweapon, okay? Culture work performed in a hood, and a worker must wear protective clothing. Class 3 pathogen, okay? It likes CO2, high humidity. There's Brucella agar with 5% serum, 30-day incubation. So all the others are like 2 weeks or 3 days. Brucella is one of the longest. It's a 30-day incubation. There's a case where I worked in a children's hospital, and there's a kid who they suspected had a Brucella infection because this kid was on a farm and he actually drank milk directly from a cow, and they thought that the baby was going to come down with a Brucella infection. Fortunately, it was negative, but we had to keep that. We had Brucella agar, and we had to keep the culture for 30 days. So hence the pasteurization. You don't want to drink. You just don't want to go up to a cow and say, I need some milk, and help yourself to it. It needs to be pasteurized. Okay. Colony 2 to 7 millimeters, 3 to 7 days. This organism almost looks like Hemophilus. So that can be a question, which organism is very similar in morphology to Hemophilus? And the answer is Brucella. Okay, almost looks like Hemophilus. So if you see that question, just remember, think about Brucella. All right, the next organism is Bordetella, and there's 5 species. Bordetella bacilliformis, Bordetella quintana, Bordetella henselae, Bordetella clarigiae, and Bordetella elizabethae. So make sure you know this slide. Bacilliformis and quintana. Bacilliformis causes Carrion's disease. So make sure you know these diseases, Carrion's disease and trench fever. And it's transmitted by sandflies. So Bacilliformis will cause Carrion's disease, and it's transmitted by sandflies. Bacillus quintana causes trench fever, and it's transmitted by the body louse or lice. Bordetella henselae, you get that from cats. This is another one that will cause cat scratch fever. But what I want you guys to know that cat scratch fever is caused by Pasteurella multocida. Okay. Bordetella clarigiae can also cause cat scratch fever, and elizabethae you can also get it from cats. So those 3 species of Bordetella you can get a cat in from cat scratches. Okay, henselae, clarigiae, and elizabethae, you get them from cats. This organism, Bordetella, is a small gram-negative rod, long incubation. It's negative for oxidase, urease, nitrate reduction, catalase. Bordetella, tissue biopsy, histology exam, silver stain, indirect fluorescent antibody, blood sample, blood cultures, subject to chocolate blood CNA. CNA usually is selected for gram-positive and incubated in 5 to 10% CO2. Bordetella automated using microscan or rapid ANA, molecular method of detecting Bordetella using PCR technique, and also 2 serologic methods. Okay, the next organism is Francisella tularensis. And Francisella tularensis is an organism that was found in Tulare County, which Tulare County is a little bit north of Bakersfield. So in the San Joaquin Valley, that's where this organism was isolated, Tulare County in California. And it's the etiologic agent for tularemia. That's the disease. And this disease is pulmonary, and it has the pulmonary and typhoid types of the infection have the highest mortality rate. So this is another significant organism, potential bioweapon, Francisella tularensis. Transmission to humans. Okay, so one group of animals I told you was the farm animals, the larger animals, the cows, pigs, and sheep. That's Brucella. Okay, so I'm throwing a lot of animals at you. The cats is the Bartonella. Okay, Francisella is wild game. So wild game is the wild rabbits, the squirrels, the game birds. So the smaller game is link that with Francisella. So if I give you an infection of a sheep or a cow or a pig, it's a larger animal, that's probably Brucella. Think along the lines of Brucella. If I say rabbit, squirrel, or a game bird, then now we're talking Francisella. Okay, blood transmitted by blood-sucking arthropods, usually ticks, deer flies, or mosquitoes. You get this organism by handling infected animal carcasses. Usually, if you go hunting and the animal is infected with the Francisella, that's how you get it. Or a flea bite from that animal that's infected. Or ingesting improperly cooked meat or contaminated water. Or inhalation of bioterror or airborne organisms. So Francisella is a bioterror, can be a bioterrorist, or can be a bioweapon. Okay. Classic tularemia associated with tick-borne. It's highly virulent in man. That's why it's a pretty significant bioweapon. Most cases are characterized by formation of slightly tender erythematous papule at the portal of entry, just like these images here. And I think that girl has it on her neck. Francisella, laboratory safety, all work with this organism should be performed in a vented hood with PPEs. Biosafety Laboratory 3. And precautions should be taken to prevent creation of aerosols and contamination of the skin. Francisella laboratory safety, recommended forwarding the specimen to a reference laboratory or state public health laboratory. Okay, so the organism is sent to CDC, use extreme biohazard precautions, and because this organism will penetrate the skin. It will penetrate the skin. Recommended specimens for processing for possible Francisella infection are sputum, gastric aspirates if you consume infected meat, gastric aspirates during systemic phase of tularemia, isolation of organisms from local lesions and regional lymph nodes. Okay, Francisella is rarely recovered from blood culture. The media of choice for Francisella, this one is glucose-cysteine blood agar. Glucose-cysteine blood agar, GCBA. Okay, make sure you put that in your media of choice. Francisella is glucose-cysteine blood agar. I'm going to be reviewing this when we do the review for the facultative anaerobes and the aerobic GNR. Colonies are pinpoint smooth gray, slight greening of agar immediately beneath the colony. Okay, mucoid drop like within 2 to 3 days. Laboratory characteristics: it's the gram-negative coccobacilles, kind of looks like Hemophilus. Cells are highly pleomorphic in older cultures, meaning that a lot of variation in shapes. Laboratory characteristics, similar simulation of growth by cysteine or cysteine. Francisella is oxidase negative, it's catalase positive, it's weak. Okay, so that's it for Francisella. Remember, Francisella is a small game, and it can be a bioweapon. Discovered in Tulare County, California. All right, ASCP sample questions. Now we're into the review. So colonies of Nyseria species turn color when redox dye is applied. Redox is combination reduction oxidation. The color change is indicative of the activity of what bacterial enzyme? Beta-galactosidase, no urease, no cytochrome oxidase, and phenylalanine deaminase. The answer is cytochrome oxidase. An example of oxidase positive, glucose non-fermenting organism. Oxidase positive, glucose non-fermenting organism. Enterobacter, no, it's oxidase negative. E. coli, oxidase negative. Club, oxidase negative. And the answer is D, Pseudomonas Aeruginosa. That's the only one of the four that's oxidase positive. All right, question, case study. A purulent aspirate, okay, so aspirate joint fluid, okay, so your mind should be thinking about a certain organism. From a 40-year-old female seen in the arthritis clinic was sent for a microbiological examination. Gram stain of the sample revealed many polymorphonuclear cells, PMNs, with intra and extracellular gram-negative diplocoxy, okay? Given the specimen type and the microscopic findings, the appropriate selected medium for primary isolation would be what? Would actually be, there's no choice. Selected media would be probably modified there, Martin. But here's a question. An important characteristic of Nyseria gonorrhea, or the infection it produces, is this an important characteristic, is A gram stain of the organism shows gram-positive diplo, right? No, that's pneumo. Asymptomatic infections are common in females. It's a possibility. Gonorrhea is disease of humans and domestic animals. I didn't talk about domestic animals. And non-pileated strains are avirulent. So the answer to this one is asymptomatic infections are common in females. All right, another question. All of the following are correct about Nyseria gonorrhea, except adversely affected by drying and fatty acids, no capsule on the outer surface, fastidious in growth requirement, serotyping exploits antigenic variations of protein. So Nyseria gonorrhea is adversely affected by drying. It likes moist environments, and also two fatty acids used as an inhibitor. Okay, and that's it for the aerobic gram-negative rods, lectures 11 and 12. So I'm going to go ahead and send this to you. There's going to be a lot to review. I'm going to put together a really big review before the review, so make sure you review it before we actually do the review. So if you have any questions, email me the questions. Homework for this will not be until the weekend before you return. Okay, so you're going to have a good spring break. Don't do anything. Make sure you do the homework and have it due by Monday on the week that you return. All right. That's it for these lectures. Again, it's not going to be included on, I didn't want to mix any other lectures with Enterobacteriaceae, and we'll just get Enterobacteriaceae this coming Friday, Friday of next week. All right. Thank you very much.